Hi everyone, I'm Tejaswee and this is Hannah, and over the summer we investigated the microbial diversity in the meadows that Conrad and Claudia talked about.
Okay, so I'm going to talk a little bit about why this is important. We're looking at bacteria in the soil, and you're probably wondering, why does that matter? So there's this thing called plant-soil feedback, and plant-soil feedback is the relationship between the plants and the soil. So basically the way it works is that if you plant a ton of one plant of one species, the pathogens in the bacteria then it creates a negative loop because those plants don't want more of the same type of plant growing right next to them because that's competition. So essentially, you get less diversity if you have the exact same type of plant over and over again. And this negative feedback eventually creates soil sickness, and this depletes the nutrients, and it's not good because we want healthy soil. So the way to work around this is by having a bunch of diverse plants, which in turn leads to diverse soil. So basically what we tested was the soil the soil biodiversity and the amount of bacteria in each of the different plots, the flowers and the flower seeds, the grass seeds, and the fallow plants. Do I just click this? So they're strongly related, strongly correlated. Okay. So basically, this is just how are we doing this? How are we testing this? Because we're taking soil samples and what happens afterwards. So it starts with the bacteria in the soil, and we run DNA analysis on it. We extract the DNA from the soil, and then we also run PCRs to actually make sure that the DNA and bacteria are present. This happens through a few ways. So, oh no, I pushed something wrong. Okay. Sorry. Okay, so we do this by we have our soil, and the soil then, there's a couple ways to do this, but the way that I'm going to explain is you can then culture the soil in like a broth, and then you can plate it, and this is basically showing us everything that's there. So we're seeing a bunch of different types of, they're different colors, different shapes. This is all a bunch of different bacteria, species, and types, and we can select one. And if we're curious about one type, we can select it and take a little swab out of it and then test the strain to see what's appearing there. And we can do that based on the isolate from soils, the colony color, the colony morphology, so basically the way it looks, as well as DNA tests. So we're testing the richness, which is the observed number of different bacteria. So how many different types are there? How many different looking guys are there? As well as, so there's 9 present here. And then that allows us to test the species evenness, which is the abundance of bacteria. So how many of them are there? That's really important because it kind of gives us an idea of, compared to everyone else, who's there. Because you might get one and it might seem cool, but if it's not very present, then it doesn't matter as much. So basically, this is just a quick overview. Diverse ecosystems tend to be more productive and more resilient, and more resistant to invasive species. So diversity is good, and we want soil diversity. So basically, we're trying to find the one that gives us the most soil diversity.
So how do we go about doing this fully in the lab? So as Hannah said, we have this one specific colony, and then we go through a DNA extraction process. So you take the swab, you culture it. After you do the DNA extraction, you have usable DNA, and then we send it off for sequencing called 16S RNA sequencing. 16S is a gene that is found in all bacteria, and to this date, this is like the whole overview of all bacteria that we know. And through the sequencing, we can put into a database, which basically for lactobacillus, this is the coding for it. So when you put it into the computer basically just spits out what bacteria you have found, and that's how we identify who is there, how many is there, and how diverse it is. So basically, you can either take it from the petri dish and do the extraction, but what we did is we went straight to the soil, and then we extracted it. We froze the soil for the second we got back to campus at -80 degrees, and then after we did the DNA extractions, we did PCRs for 16S RNA sequencing. We looked at the richness and the evenness to see if plant diversity affects microbial communities within each of the treatments. So the process that we had was we have all these plot size, mix A, mix B, and fallow, and we randomly did coordinates for our sampling because we didn't want there to be bias as to where we decided to pick our samples. So we did random number generator. We collected our samples with these corers. So here I am with a corer in the summertime going into the plots, and then we collected into those little collection tubes, and then after we did DNA extractions, PCRs, then send off to sequencing, and then finally, for data analysis, we use R programming to see who is there, how many is there, and the diversity overall.
Okay, so this is just the bacteria that were present. We had over 20 different phyla, so each color is a different phyla, and then there was over 20 phyla present in each species, not in each species, in each sample. So you can kind of just see there's a lot going on here. It looks pretty similar, but as you can see, some have some colors, some don't have those colors. It's a good figure just for kind of seeing the diversity, but it doesn't give us a lot, so we're going to go a bit more into that. So this is the big takeaway. Plant diversity affects microbial diversity, and most importantly, Mix B was found to have the least diversity overall. So Mix B was the grass mix, and the fallow and Mix A, which was the flower mix, were found to have the most diversity. So, well, the most significant diversity compared to Mix B. So what we're taking away from this is that fallow and Mix A are giving us more, and we want more diversity, so that's good. So fallow and Mix A worked really well. Mix B was a little bit less successful, but we'll talk more about that.
So another way we look at microbial communities are ordination plots. So if you look right here, fallow and Mix A are very similar with the amount of bacteria that is present, and whereas Mix B is more away from everyone. And this is very significant because fallow is just letting the grass run wild, whereas Mix A, you buy the seed, you have to spend money on it. So honestly, letting your plots run fallow is cheaper and gives you the same amount of diversity. And if you look at panel B, we're all just comparing fallow to Mix B, because since fallow and Mix A are very similar, you can only compare two analyses on this. So these are all the bacteria that's present in fallow, but only 6 is present in both fallow and Mix B, whereas Mix B is missing 26 bacteria. Overall, there's 32, but they only have 6 in common, so there's a big difference as to who is there and how many is there for bacteria-wise. So going on to, there you go. Going on to why we have what we got with our data, antibiotic pollution is a really big thing, and we also decided to look at antibiotics and antibiotic-resistant bacteria within these plots. So because of hospitals, the general population, agriculture, aquaculture, and the industry, we're polluting our environment very much, and it's affecting our soil. So because of our pollution, we are becoming more resistant to bacteria and creating more antibiotic-resistant bacteria within our soil, within our environment. So we decided to see if these treatments would have different effects on this gene called INT1, which is a marker for antibiotic pollution. But not only that, it also affects pesticides and herbicides and everything else, so it's a very great marker because it's in all the genes that have antibiotic-resistant bacteria, and also they have a really big rapid generation time because of this thing called horizontal gene transfer, which basically allows different bacteria to share genetic material. So basically, this one bacteria that is not resistant can share bacteria with a resistant gene and then confer, allowing them both to be resistant. And we don't want that. So, as I said, all these different things are causing us to pollute our environment. And because of this, we also looked at transect sites with Conrad. So we had 4 different transects near the stream along Farm Hub, right next to the stream, and then 300 meters away, and then 600 meters away. And Hannah's gonna talk more about as to what we found with INT1 and antibiotics within our normal plots, and then those transect sites.
Okay. There it is. Oh, okay. Our graph did not come in, but that's okay because I can tell you the graph. So, so the first graph is basically of the plots. So the plots were Mix A, Mix B, and the fallow. So between those, there was very low INT1 presence, and there was nothing significant there, no significant differences between the plots. And it was also very low, so it's not wasn't anything exciting there. But one thing we found that was exciting, and Conrad, I'm really excited to tell you too, is that with the transects, we found that the ones were closer to the water. So there's a little stream near the transect sites, and it's next to the water, 300 meters, and then the 600 meters. And with those, we found that the ones that were closer to the water had higher int1 than when you were further away from the water. And this is pretty consistent with what we'd expect to find, particularly because water tends to have int1 present in it in all streams and just from runoff and different things. So that's pretty consistent with what we'd expect to find, but it was really interesting to see that gradual effect of the distance from the water. And with that,
we would like to thank Bard College and the Summer Research Institute, Farm Hub, Conrad, Claudia, Anne, and our class Bio 340, which is helping us further our analyses with this farm plots. And thank you.
Thank you. Questions?
Just a quick question. You said mix A was grass, and then what was mix B primarily?
mix
Mix B was grass.
Yeah, mix
What
B
was the
was
other
grass. Mix A is a wildflower mixture.
one? Okay. All right, thanks. I just missed that. Sorry.
And at the end there, were you saying that the bacteria closer to the water source had more of the antibiotic resistance? More or less?
It had more. So it had, it had more present closer to the water because that water sources that have runoff in them and where wastewater treatment eventually goes through does tend to have more antibiotic resistance present in it. So that's pretty consistent with what we'd expect to find.
So, I'm interested in nitrogen cycle in the soil. So, some of the bacteria that, identified, listed, plays a role in the nitrogen cycle. But were you able to quantify, what percentage of bacteria that usually, play a role in the nitrogen cycle in the soil? Because that also dictates, the health of the soil for plants.
Yeah, that's not what we have done so far, but Gabriel, I'm sure, has more ideas to how they all play a role. We also have another professor, Kathy Collins, who specifically looks at the soil, so I'm sure Gabriel and Kathy have something going on, but for us, it's just what we know so far is not anything with the nitrogen cycle. I'm sorry.
That's fine. Thank you.
Of course.
I was just wondering if you guys, I'm really surprised about this big difference between A and fallow versus B. And did you guys have any discussions with Gabriel that would maybe have some hypothesis as to why the grass-rich plots had so much less bacteria?
We don't really know that much about why. This is just new data that we only got back the sequencing very recently, and then after that Gabriel left for Oxford, so we didn't really this is all just on us. We are gonna talk to him once he's back for sure, but we don't really know why. But it's significant because mix A you're paying for it, and fallow you're not paying for it. So that's the only biggest thing that we have.
I would also note that with flowers, you are getting pollinators, and that can encourage sometimes different growths of different nearby flowers, whereas grasses don't give you as much to go with, would be my guess.
Also, with the insects, Conrad, you also got not a lot of insects in mix B, whereas mix A, you got the beetles, the spiders and stuff, so I'm sure the insects also brought a lot of diversity into it.
I'll talk to you about mine.
I found that really curious too in regard to the fallow in both Conrad's presentation and in your presentation being, in a sense, coming out ahead of even the wildflower mix. And I'm curious to know if that potentially has to do with the fact that the wildflowers that were coming in, I forget which ones you mentioned, I think fleabane, some of the wildflowers that were coming in naturally in that area versus your wildflower mix. If there was a way that perhaps you could then go and study in the fallow sites, like what were the particular species that were coming back naturally in the area, and then purposely planting those as part of the wildflower mix as opposed to just sort of a general regional mix, because that specific, in a way, to me, it speaks so much to the specificity of the particular field, even within a particular region. I don't know if you could speak to that, either of you guys.
I think your plant mix was, you were saying that it was like specific mostly to like the where we are. So I think it was, yes?
It was mostly plants that we know from our environment in the Northeast here, but there were some Midwestern ones that we invited into. And I guess what you're saying is true, but we should also not forget we did have a lot more native bees and a lot more butterflies in the flower-rich species mix. So I don't think it was bad in all regards, but it is interesting to see, wow, the fallow is actually not so bad in some respects. Yeah.
Hi, thanks for your presentation. So I'm thinking you tested for bacteria, and so just thinking about all the different microbiome, the life in the soil, it's not just bacteria, and maybe the fuller picture of the biodiversity is if you're testing for fungi and other microbial life. So because the little that I know, just limiting the looking at the biodiversity through bacteria is limited to that world of soil, of the soil life. So it's kind of confusing to me, like in Elaine Ingham's work, which is a little bit of work that I know, she talks a lot about the different types of microorganisms that will inhabit the soil depending on what you're growing. And so the bacteria would be dominant in a fallow field where you have annuals, and bacteria is more found in annual production versus the grasses, I think, are perennial grasses, and they're going to be more fungal, or other microbial life will dominate those soils. So the biodiversity, just looking at bacteria, is confusing in my mind because there's a lot more in the soil to look at. So to judge the biodiversity just with bacteria perhaps is not as illuminating.
Yeah, no, for sure. We have a lot of limitations to our experiment because we only also are able to take off 2 inches off of the top of the soil, because when you go deeper in the soil, there's obviously more bacteria, more fungi, a lot of other present in it, but we didn't want to disturb the entire plot at all, so we just did as much as we could, and just off of the top of the soil, that gives us a lot more information than we had already known before. So we know that there's a lot of limitations, and we hope to expand our experiment further on. Is there anything you want to add?
Yeah, and I think it's also just important to note that bacteria does give us a pretty good sense of what's going on because there's relationships between the bacteria and the fungus. And what we know is that diversity of the bacteria helps the plants. So we know that's beneficial, and we know that relationship with the bacteria and the fungus is mutual and exists for both. So I think it's important to realize that we don't necessarily work as much with the fungus, but it does still have a similar effect of the diversity being important and having a role in being beneficial for the plants. But yeah, nutrients and fungal would be a good one. I think a lot of farms do test the fungal bacteria ratios.
I think you mentioned that there were 16 bacteria species that you found in Mix A, or in for the fallow field, but not in the other, in Mix B, is that correct?
Yeah, let me pull up the graph for you. So over here in B, there's 26 that are in total, and then there's only 6 that fallow and Mix B have together, whereas all those that you see away are what fallow has, but Mix B does not.
Did you have any ideas or plans to investigate why, what makes these 16 species of bacteria?
Yeah, that's something that this is new data that we just got. It's just on diversity. It's just the fact that fallow allows these bacteria to grow and prosper, whereas Mix B does not. And we just have to test a lot of other things to know exactly why these species like to inhabit fallow versus not Mix B. You know anything?
It also ties a little bit back to the plant soil feedback. So as we had talked about earlier, when you have more plants present, we'll get way back there. When you get more plants present, that creates more bacteria present in the soil. So the way it works is these, let Do I have any good ones? So there's mutualists, which are the little circles over there. And then there's pathogens, and when you have a ton, you get a bunch of different ones versus just if you plant like a whole field of like roses, you're gonna get specific pathogens just to roses. And then when you try and grow roses there again, it's gonna be a lot more difficult. So we're probably seeing that because with mix B, there's less diversity going on compared to the fallow plot. So when there's less of it, there's less mutualists and pathogens growing as well. Does that kind of make sense? It's the relationship between the two.
I don't know if I heard it and forgot it, but were all 3 plots tilled the same amount of times at the same times? Okay, thank you.
All right, thanks very much.